All right, we're back in the canola plots, so we're going to talk some canola diseases, so specifically sclerotinia, blackleg, and a little bit on clubroot. So it is very dry, but we're doing our best to find something to show you guys. So what have we been able to see in terms of sclerotinia so far?
Yeah, so first let's just talk about sclerotinia risk.
Sure,
right?
yes.
So I guess the first thing is this crop is outside of the recommended fungicide timing. So our window is 20 to 50% flower. If conditions are favorable, you're going to want to be on that earlier side, so that 20% flower, which we're way beyond here. And the reason for that is if we get infection at that stage, we're more likely to get the main stem infection. So that's going to contribute most to yield.
Okay.
And so we know that the sclerotinia pathogen is kind of a weak pathogen. It uses the petals or senescing tissue as a nutrient source. Those petals then fall within the crop canopy, come in contact with the leaf, and then cause infection from that. So what we're seeing here is, again, we had to dig pretty good for this, but we're seeing areas here where a petal likely fell onto the leaf, became attached, and then caused infection. So we're seeing some chlorosis. You can actually see the outlines of the petals here. So that's the initial stage of sclerotinia. Then the pathogen will grow into the petiole here and into the stem and cause the lesions that we're seeing. So these were on the main stem, so these would likely result in main stem lesions. But if they were on the branches, that's where we get those branch infections, which likely occur later in the season. The other thing that we're seeing is that we can see here that the petals are falling onto the leaf. So that's where they're going to come into contact, and for infection to actually occur, this leaf would have to be fairly wet to cause that petal to actually stick. So this pathogen really likes moist conditions, a high humidity and moisture, which we're not seeing in here. We walked right through all these plots. Our plants are still dry, giving us an indication that's not the highest risk for sclerotinia development. So that moisture is going to be important right from the formation of those little apothecia all the way to actually that petal sticking, that initial infection, as well as that infection within the canopy to actually cause the lesions. So I'd say this crop is fairly low risk for sclerotinia, or these plots.
Great. I should also just mention, feel free to ask questions this whole time, guys. I can lead a discussion, but it's much better when you let us know what you actually want to hear about. So the next disease, blackleg.
Yeah.
So how do we scout for blackleg?
Okay, so there's two different times you can scout for blackleg.
Okay.
So first time to scout is early in the spring. So looking at when that canola crop is first emerging. So we're going to be looking at the cotyledons and the first leaves for lesions. So lesion can look like a little chlorotic spot or with a tan center, and we're looking for the little black pepper-like structures or pycnidia within that lesion. So that's usually, so if we get infection at that cotyledon stage, that leads to that main stem infection, which is what's going to cause the cankering or that black discoloration. So that's going to be the highest contributor to yield loss when we get that early infection.
Okay.
But the best time to scout for blackleg is actually later in the season. So when you're swathing or at potting stage, to go and pull some plants and actually look at what's going on. So this information is going to help you guide your rotations and understand how your management practices are holding up. So when we pull this root or pull this plant, we'd want to be cutting about here. And so what we're looking for is actually, we'd be looking within that tissue and looking for blackening within that internal tissue or the pith, which is usually white. But while you're doing this, it's also a really perfect time to look for clubroot.
Right.
So any time that we're in a field, canola field, we should be pulling plants and looking at what's going on in the roots. Pull plants at the field entrance as well as low spots. And so we'll be looking at the roots here. Under kind of early infections, which is where we really want to find clubroot, is you're going to see small swellings on kind of the lateral roots. As the severity increases or the amount of the pathogen in the field increases, the gall or the swelling is going to become bigger and may actually occur on the taproot and can take over most of the root. The earlier we find it, the easier it is to manage.
Okay, and so I know we don't have pictures of some of these diseases, but if you sign up for the Ministry of Agriculture's newsletter, Crop Production News, this will all be in next week, so you'll be able to see pictures of all of these diseases. And also, we had a question earlier this week, and this is probably a good time to talk about it, but somebody asked where we get our booties.
Yeah.
So why do we wear them, first of all?
Exactly. So the booties or the boot covers are a really good step in terms of biosecurity. So we work into many different producers' fields, so we want to make sure we're not bringing anything into that field, so including soil-borne diseases like clubroot, but it can also prevent moving things like Aphanomyces or Phytophthora root rot. So it allows us to have a barrier between our feet and the field, and then we can take them off in between. And where do we get them? That's always a really common question. So there's a few different places. There's lots of companies that offer them. There's lots of styles. There's these blue ones, they come in clear white ones, they come in Tyvek ones, they come in short tall ones and tall ones, anything you would really desire. So you can get them at Uline, but also VetSource Canada has a variety of them as well.
Excellent. So if there are no questions online, we will move over to some cereal diseases. So see you guys in a couple.